to the last talk, which is mechanisms of resistance to treatment in pulmonary fibrosis. And Oliver Eichelberg from Munich will present the lecture.
Thank you, Chairman, for the introduction. I want to thank the organizing committee, in particular Leo and Enrico, also for inviting me here. Fantastic meeting. I thought this is probably one of the first times that the weather here is worse than in Munich. But the dinner made up for it clearly. So I want to follow up on what Guy and Ian actually have outlined so beautifully in the previous two talks. So one is I try to convince you that while you think we're talking about lung fibrosis, particularly idiopathic pulmonary fibrosis, that this is an interstitial disease, there's evidence coming out more and more that this is also an airway disease. And I'll show you data to that, and I try to convince you of the role of the airways actually in IPF. And secondly, that also probably will go along to what has been said, and that is, A, we're at the verge of really a scientifically biomarker-guided therapeutic approach, and B, it needs to be symptom-driven. And again, the role of guidelines has been addressed, and that applies to this topic as well as to the others. These are my disclosures for your information, and this is the disease group we're talking about. So we basically will focus on idiopathic pulmonary fibrosis. Here is the whole group of diffuse parenchymal lung diseases, many of which are quite rare, some of them are not so rare. We're talking about the idiopathic group of idiopathic interstitial pneumonias, and there we talk about idiopathic pulmonary fibrosis. Why? Because this is the best characterized group. We know the most about this group. There have been recent, quite substantial advances in our management of this group, but it's also still the most deleterious disease that we have in this disease group. So let's zoom in on the disease from a helicopter view and then going into the detail into the lung and into the particular areas of the lung. This is just a brief outline of how the lung looks. Many of you do know that. On the left side, you have a normal lung. On the right side, an IPF lung. The IPF lung is characterized primarily by a very heterogeneous pattern, and that is spatially heterogeneous. You have very normal areas, close to very fibrotic areas, close to so-called emphysematous honeycombing areas. And that can change from patient to patient, but also from locus to locus within the lung. So the heterogeneity is really one of the prime characteristics of the disease. On CT, it looks like this. Again, you can appreciate the heterogeneity. Stefania has outlined some of these patients yesterday in terms of early disease, and this applies to late disease as well. If you zoom in into the histology of the disease, you see this on this slide here, a normal lung, which you're all familiar with, and here an IPF lung. And just to mention one thing already, is while we always talk about extracellular matrix, interstitial fibrosis, which you can of course see here, and I think we all agree that this patient has difficulties breathing, you can also appreciate the numerous appearances of so-called bronchialized airway structures here, all across the lung, basically that we don't see in the normal lung. We don't see these structures. And current biopsies actually, most of the characteristics of the biopsies actually go along with this feature. If you were to stain for an extracellular matrix protein, this is collagen, this is how the lung looks. There's massive infiltration of extracellular matrix around the interstitium, but also around the airways and vascular structures. So just to recap and highlight the principal components, one, we have the interstitial matrix and the so-called fibroblast foci. So these are the classical hallmarks of IPF that we all know from the literature. But second, more and more we have these distal bronchialized structures that are appearing in the disease that are either originating from alveolar space and hyperplastic alveolar epithelial cells, but more likely from disturbed bronchialization and bronchial epithelial progenitor differentiation. I'll show some data about that. And these are the highlights or the histological hallmarks of IPF compared to a normal lung as indicated here. Now, for our research and for understanding the disease, we do need tissue biopsies. It has been highlighted before. Clearly, this is the stronghold of our field, respiratory medicine. But in IPF particularly, this is changing currently, and this is outlined here from one of Ganesh's latest articles. This is the guideline we all adhere to currently. And the one thing I want to highlight here is that surgical lung biopsies are really not required in most of our patients anymore. So the availability of tissue is limited now and in the future even more. Because for most patients, if you have clinical picture and imaging by HRCT, so the radiologists and the clinicians agree, then you have IPF here. There's no need for a surgical lung biopsy. And that is a problem actually for some of the patients, but mostly for research and understanding the disease. The landscape has significantly changed, and that is probably also why the topic was chosen when we have 2 approved substances that were introduced to the treatment of mild to moderate IPF. And these were one from Roche, one from Boehringer. We all know these substances. That's about 1.5 years now where we have extensive expertise with these compounds. And again, just to recap what has been shown yesterday, these compounds basically slow down progression. That's all they do. And here is pirfenidone. You see the decline in lung function is slowed down a little. You don't really change disease course, but you slow down progression of disease as measured by lung function decline. And this figure has been shown yesterday from Luca's papers. Again, the same holds true for the Boehringer compound. So what they do is they slow down progression of disease. So when I got the topic, I then got very much excited because it was a talk you usually don't really give so much. So you go to PubMed, as we all do. You type in resistant to treatment and lung fibrosis, and out comes no items found. So thank you very much for your attention. But let me turn it around a little. So let's just talk a little bit about the opposite. Resistance is the one thing, but susceptibility is the other thing. And I think that is something that has really changed the field of IPF, but also lung fibrosis in the past, I would say, 3 to 4 years. Our understanding of susceptibility has massively increased, and we're at the verge of entering this into the clinical picture. In many centers, not only centers of excellence, but I think this will also appear in the general practitioner's office sooner or later. So here's a classical disease trajectory. And again, this applies to everything we talk about, not just IPF. It applies to COPD, it applies to asthma, any lung disease you picture, actually any disease you picture. We know that upon diagnosis, many of our patients go on different trajectories. It can either be pretty stable or slow declining or rapid decliners. These trajectories are all complicated by so-called acute exacerbations. The definition of which, of course, is different between IPF, COPD, and asthma, and that is actually something that limits the field of exacerbations currently. And this will then basically complicate the trajectory, or you can also switch from one course to another, and you have the same rapid decline. The second challenge we have, we diagnose all of our patients too late. So in IPF particularly, diagnosis is done very late, mostly in the 5th, 6th decade of life, and we don't know what's actually happening here, as opposed to asthma, for example, which we know very well from childhood asthma. We don't know the early signs of fibrosis in our patients before we diagnose it. But let me give you, and let me try to convince you of sort of a blue sky vision in IPF. I think that in the near future, 2 years, 5 years, 10 years, definitely the nearer future, we will actually be able to have a very rapid and distinct and definitive diagnosis of interstitial lung disease. I think we will be able to predict the disease course, what I just showed you for the trajectories. I think we can individually assess bioactive and druggable signaling pathways, and as such, we will be able to predict therapeutic responses or even engineer personalized approaches. And one thing that I'm going to introduce that is on the verge of entering the clinic is genetics, and I think genetics will pave the way for these points. So let's review the early IPF genetics for a second. Many of you will be familiar with some of these papers. Originally, the very early observation that in some very rare cases of familial fibrosis, you have SBC mutations by the landmark paper of Larry Nogi. That was 2001, so 15 years ago. From then on, we had multiple confirmations of rare mutations in SBC or other surfactants, for example, here SBCA2. And then the story of the telomerase complex-associated protein STIRT that has been originated from Mary Armagno's work. So these were rare mutations in familial cases of IPF that actually showed that indeed lung fibrosis is something that can be genetically inherited. But the real breakthrough happened when we actually found that in familial pulmonary fibrosis, but also idiopathic interstitial fibrosis, we actually have genotypes in the MUC5B locus here that would actually be highly enriched in our patient populations. And I'll review the data for this quickly. This is the most important paper, I would say, in the New England Journal by David Schwartz's group that has actually shown that there's a common genotype, a common promoter polymorphism in the MUC5B gene that is significantly enriched in patients with idiopathic pulmonary fibrosis. And this is the immunostaining of the MUC5B protein, and you can already visualize that actually this is a protein, as we know, that's secreted into the airway lumen, so already pointing a little bit toward the airway as a prime compartment of disease origin in IPF. The locus was found by a genome-wide association study from the same group, David Schwartz here, and here is the classical Manhattan plot from that original study, and you can appreciate the linkage and the significance of linkage here of MUC5B right here. It is the top gene linked with IPF. And actually DSP, we've just published in the Blue Journal, also from David Schwartz's group here, a paper that shows that DSP is also linked with fibrosis. So that is something that is emerging more and more and is confirmed across the world in multiple cohorts, actually more and more. The importance of that genome-wide association is shown here. Obviously, across our diseases, we have tons of genome-wide associations in pulmonary research, but many are non-functional. You just have an association, but it doesn't really reflect a different product, protein product. It doesn't really reflect a different function or expression. In most of the cases that I'm going to talk about with IPF, these are all functional polymorphisms. So that is shown here in the same paper, the Nature Genetics from 2013. Here you have controls, and here you have case IPF. First of all, in cases, expression is highly enriched. Note that this is a log scale. And then also it's highly enriched with respect to genotype. Here are most of the minor alleles, and you see it's higher expression, i.e. indicating that these are functional genotypes. Let's review the facts quickly here. So this is the polymorphism, the SNP. It's 3 kb upstream of the transcription start site. It has a frequency of 34% in cases of familial interstitial pneumonia and 38% in idiopathic pulmonary fibrosis. 9% among controls in the original study, but I show data across the world. And these are the odds ratios for heterozygous and homozygous. So it's important to note that this is the best risk factor for IPF that we have by length. If you look at the odds ratios of 20.8 or 21.8 for the homozygous minor allele right here for familial and here for idiopathic pulmonary fibrosis, this is the best risk factor by far. This is massively increased and a massively higher risk factor as many of the other risk factors we have in all sorts of respiratory diseases. Here's the conundrum. If you have that risk factor, if you have the minor allele, the TT, you have a much higher chance of developing IPF. But if you have IPF and you have the minor allele, TT right here, you likely have better survival. We cannot explain this. It's important to note, however, that at this point, this JAMA study is the only study that looked at survival in terms of genotype. Multiple ones are going to be coming out now. But again, this awaits replication. But at this point, increased risk of developing IPF. But if you have IPF and minor allele, you have less severe disease. Important that this finding has been replicated across the world in most populations except one, and I'll quickly review this. So here this is the paper again from the Denver group with the Mexican population and actually incorporating the data from Moises Selman in Mexico City. Again, increase is associated with IPF in this population, but also here, the authors note that the Korean population they investigated here actually doesn't have that genotype, and that has also been replicated at least once right now. So it's increased all across the world, but not in a Korean population. Here are some of the papers that have confirmed this. So again, Japanese population, same. In the European Caucasian population, the same, and also Chinese, but not in the Korean population. We don't know why this is the case, but apparently there seems to be some genetic spectrum in Koreans that is not associated with the MUC5B locus. Here's the allele frequency of that TT locus in the general population, and you see this is from the PLOS-ONE paper of 2 years ago. Chinese is very little, so they have more increased odds ratios, but French, Italian, British, American, all around 10% in the general population that was investigated. Note that the numbers here are not so great. These are ranging somewhere between 300 to 500. This is way too small to read for you, but this is the German analysis of population-based cohorts. Now here we're talking about 2,500 individuals, and again, the allele frequency in Germany is about 7%. So we've measured that extensively, so we in Germany are somewhere in the middle, located here. This is our prospective data from our Munich cohorts for interstitial lung disease and COPD as controls. Again, it's important to emphasize that we routinely, prospectively measure genotypes for all our patients at this point in time. And again, here I show you data for 125 IPF. The numbers are up here, and 195 non-IPF ILD. So this is incident cases within a year. So this is a 2015 compendium here. And you see, indeed, if you look at all the genotypes here, this one stands out. This is a MUC5B, minor allele, highly enriched in the IPF population, not in the non-IPF ILD, as a control population. So this is something that is very robust and solid. Now, if we want to understand now how this genotype is actually leading to disease or is actually inducing disease, predisposing for IPF development or fibrotic transformation, we of course need tissue to understand that. And I showed you before our blood profiling, genotyping, and this is now profiling from all our transplant patients from the same year that we were able to obtain. So here you see now lung DNA extraction, so whole lung homogeneous. This is 30 cases. Again, here you see IPF, highly enriched for the heterozygous, but you're lacking the homozygous minor allele. This is very interesting. This is by no means statistically sound, but apparently, indeed, if you have IPF and the minor, you have less severe disease, and we have less patients that are transplanted actually with the minor. In fact, we have no patient from the whole year of 2015 and 2016 up to now that has a minor homozygous allele that was transplanted. You derive cells from the lungs to functioning analysis, and again, this is a small number, but it shows you the same thing. There's no minor for functional analysis, but we have the heterozygous genotype for functional analysis. Now, is this evident, and does this only matter for a very small population or just for our IPF population? So let me show you some data from a very recent paper from Matt Hanninghake that actually looked at interstitial lung abnormalities in the Framingham Heart Study. And you can see here, if you profile the genotype in that study population, and you look at the minor allele, and you correlate minor allele with the absence of interstitial lung abnormalities. So again, this highlights what Stefania has outlined yesterday. Presence of interstitial lung abnormalities and definitive fibrosis. You see there's a significant enrichment of these two phenomena here in the case of the minor allele. So indeed, this applies not only to a very highly select IPF study population, but it will apply to the general population. And in fact, in Munich, we're actually screening all our population-based cohorts and follow up our population-based cohorts with the minor allele for interstitial lung abnormalities now. It's important to note that it has been looked at extensively, whether this only applies to IPF. And indeed, this seems to be the case. Here's just one study highlighted from Ian's time as chief editor that was published from the London group here, that indeed it applies to IPF, but not patients with systemic sclerosis or other forms of interstitial lung disease. At the end of the day, we have to ask ourselves, what is this MUC5B protein doing? There's one very nice paper published up to now, and this is again from the Denver group. Chris Evans is the lead author here, published in Nature 1.5 years ago. And it shows indeed that MUC5B primes innate immune defenses, which would make sense. Importantly, it primes defenses in the airways, macrophage phenotypes in particular. So again, coming back to the airway as the select compartment for driving disease pathogenesis in IPF. Now, does this also apply to being relevant in a clinical study setting and actually a therapeutic response at the end? So is there proof or is there hope that indeed, depending on a certain genotype, you will have a different therapeutic response to the two drugs that we know, but also maybe to other drugs that are going to be coming out sooner or later in IPF? This is a controversial study. We did publish this in the Blue Journal last year. I'll show you the reasons why this is the right decision, but I also point out the flaws, and Guy and I have already discussed that yesterday in the lobby. This is a paper from Justin Oldman. It's a retrospective analysis from an NIH-sponsored trial. Now, you know that in the PANTA trial, there was no difference in terms of decline of lung function here between NSC, acetylcysteine, or placebo. Zero effect. That's why the trial was also stopped at the end of the day, because there was no therapeutic beneficial effect here. This is from the New England Journal paper in 2014. Now, when the authors retrospectively looked at the data from the PANTA trial and actually genotyped the patients and segregated now not for the MUC5B, but again TOLIP genotype, which also was found in the initial study here, And they looked at the genotype and the therapeutic response towards NAC. This is what they found. So depending on the genotype, and here you have CC and TT, the therapeutic response was totally opposite. In the one instance, you had a beneficial therapeutic response. In the other instance of a genotype, you had a therapeutic response. So actually, you get worse. I think we were right to publish this study in the Blue despite all the deficiencies of the retrospective study design, post-hoc analysis, small numbers here. But this is the first study that shows that genetic profiling in interstitial lung disease may be a very important factor for profiling therapeutic responses in the future. And I think it's time to do that systematically and prospectively. So here you have the conclusion from the paper again. Again, if you have a certain genotype, you actually do harm with certain therapies. And we know that this is the case in other trials in IPF currently, that you have adverse effects, and that may also be due to genotypes that are prevalent in that population. So take-home messages from this part of the talk. IPF is associated with increased abundance of SNPs, genotypes in the MAC5B and TOLIP genes, but others will come out in the next year or this year and the next year. DSP is one of them. Genetic disposition, as measured by these genotypes, may predict treatment responses. There's good evidence, at least in one retrospective analysis, that this is the case, await screening in prospective trials. But I think it's a time where genetic screening should be performed in all clinical and experimental studies and address the contribution of these genotypes. I think that it doesn't make sense to not do this anymore because the science tells us differently. Let me conclude with one nice observation, or again another observation that pinpoints the airway as one of the key sites in IPF. And I briefly go through this. Now this is unpublished data. This is again the histology. Here are all these bronchialized lesions, these airways that are definitely different than the normal lung. And you see, this is a picture we often see. So we actually asked the question, what about this elusive lung stem cell? You probably are all aware that there was a New England paper a long time ago, 3 years ago, Piero Anversa's group, claiming that c-KIT positive cells are the multipotent airway stem cell or lung stem cells in humans. Has not been confirmed, has actually been refuted. But we are still on the search for a progenitor cell that actually is disturbed in disease in the human. We know a lot in mice, but the human lung is so different, and it actually demands investigations in human samples at this point, I think. So here's an airway, classical airway from a normal human lung. In red, you have P63, that's nuclear. These are the basal stem cells in the airways that are thought to be the progenitors, actually. And in green, you have keratin 5. And you see some keratin 5 stains, positive with P63, double positive, and some not. So these are presumably two different populations of progenitor cells. That has been very nicely outlined in the mouse lung, but very controversial in the human. If you look at the healthy human lung, and now this is the work by a postdoc that has joined the lab, and this is tedious work for a long time, and Marina and Manuel know how tedious the pathological examination and quantification thereof is, actually, of histological samples. And she profiled actually single positive and double positive populations. This is the key of the study now. We don't know about numbers of these stem cell populations in the human lung. So here you have conducting airways, large airways, smaller airways, distal airways. You can immediately see here you have a lot of three colors, yellow, green, red. So you have single positive and double positive populations. Here you only have green, only single positive populations. She quantified this here per millimeter basement membrane, and you see again in the distal airways you don't have in the normal lung these populations of keratin 5, 14, P63 double positive cells. This is the IPF lung, and again we've looked at a lot of IPF lungs now, and this is the distal lung, and again you see something that you don't see in the normal lung. You see three colors, green, red, yellow. So we have the IPF airway, in the IPF distal airway and actually interstitium, cells that exhibit potential progenitor function, and we don't understand what they do. Importantly, if you stain this with differentiation markers, one population, the classical one, keratin 5, co-stains with all differentiation markers. Here you see a lot of yellow. The other population, keratin 14, never co-stains. So here we have a population of progenitor function that clearly primes the IPF lung for some sort of disturbed basal or airway epithelial differentiation. And we think this is very important, and actually it plays very much into the story of the genetics, highlighting the airway as probably the site of origin of the IPF lung. And this is my last take-home message. keep in mind that in the future, it won't be so much the interstitium anymore in IPF. It will be the airway, and it will be misguided differentiation of airway epithelial cells. And I leave this, so I won't talk about Viagra or sildenafil. I talk about hair in older populations. That applies to me, myself as well. We're now taking all these epithelial progenitors, and the nice thing is you can differentiate them in air liquid cultures. Guy has already shown data from these sort of investigations. And this is a picture I leave you with. So here you have ciliated cells and non-ciliated cells. Beautiful hair. You can actually, our airways look like this if you look onto them. And this is Teotihuacan. This is a beautiful visit we did to Mexico last year. And those of you who have been there, it's a fantastic site. 50 years ago, these pyramids were all covered. Nobody knew that there were pyramids. If you stand on the pyramid de la Sol or de la Lune, this is the de la Sol, stand up here, you look around, you see so many hills that all seem to have pyramids underneath them. And it's such an amazing sight to see how little we know, even on our Earth. And I think the same applies to IPF. We're at the verge of standing off one of these sites. We have looked at some phenotypes, we have understood some phenotypes, but I think in the next year it's going to be an exciting time that actually will translate to the clinic in IPF research, and I'm very happy to be part of this. This is the lab that has done most of the work. These are fantastic international collaborators, and I thank you now for your attention.
Thank you very much, Hovel, for this stimulating lecture. Before opening the floor for discussion, I should like to ask you, in your vision of the blue sky for the future, what role could have a selective inhibition of fibrogenic factors and of signal transduction pathways that they induce? I ask this because we found that nerve growth factor is overexpressed in the lung, with the fibrotic lung, can induce a differentiation to mucus metaplasia and influence the production of MUX5AB, and also the activation of myofibroblast. As you know, nerve growth factor acts through a high affinity receptor which induces tyrosine kinase activation. Also, this is one of the factors. Of course, there are other fibrogenic. Do you think that this can be an approach to go together with the genetic influence?
Absolutely. But it will be an approach, I think, practically that will be limited to select group of IPF or lung fibrosis patients. And again, I would like to avoid the term IPF actually at this point, because as Ian has outlined, I think it's symptom. And you know, in all our diffuse parenchymal lung diseases, there are similar symptoms and different symptoms. So the therapies we have will apply to a lot of different other disease groups other than IPF. But in those select groups, for example, where you can measure bioactivity, as you outlined, for certain factors or others, and my bias would be you need lavage fluid. So this is a proof of principle study that we've finished about. You can measure in bronchial lavage fluid, you can measure bioactivity of certain factors. And you can measure actually on-target efficacy of treatments. So if you get lavage fluid, you're able to do that. If you don't get lavage fluid, I think we're a couple of years away from doing that in an exact manner, because in the peripheral blood, we cannot measure many of the pathways that we can measure in the lavage. But in those study populations that are subject to repetitive lavage, before, after treatment, I think you have a very rational approach to do exactly what you mentioned.
Thank you for the presentation. It was, very interesting, and just lovely to see somebody looking forward for differences in the disease, so complicated than that. One thing that impressed me, it was, that you show these beautiful pictures of the keratin, and the epithelium, the new cells and so on, that they only seem to be occurring in IPF. Interestingly enough, I was kind of, this is exactly, almost the same picture that is shown in the New England Journal of Medicine pictures of the MUC, kind of promoter and so on. Is there any relation? Have you kind of been looking in a possible relation between, you know, these two different things that you are seeing on the airways? And
So we'd love to correlate this to the MUC5B status, but when you remember I showed you our profiling of our cohorts, we don't have minor in our transplant patients. So actually we can profile together maybe your cohort and see if you have actually minor alleles, because you can also sequence from the slide by just scraping. But in Munich, 0. In 2 years we have not one minor that was transplanted, and we do cryobiopsies, but the quality of the cryobiopsies for this staining is you cannot quantify really. So we'd love to look at bigger cohorts, and then of course if you had a significant number of minor, MUC5B or TOLIP, it would be nice to correlate, absolutely.
Please.
Do you have also an idea about predicting the benefit of NAC treatment in patients with COPD?
Well, yes. You know, I mean, the COPD field, but again, Guy is the guy who should answer this question. You know, the COPD field and the genetics and COPD gene and the gene profiling has not been so black and white as an IPF. You know, we started out in between our different, fields in respiratory research, and IPF was always thought, oh, this is the burnt out scar. You know, nothing is happening in this lung. Turns out now, replicated all across the world, if you have a piece of lung tissue, the IPF lung is the one you can diagnose by the signature, and it's actually characterized by an onset of all the developmental pathways that we know from development. So IPF is not a burned out scar, but it's a highly active disease, as you see here, that is actively, proliferating, differentiating. And COPD, but again, Guy, you may want to add, and COPD is a lot more difficult to tackle in terms of that activity profile. You don't have so specific markers, I mean, IPF, 3 lungs were enough to find MMP7 in all IPF lungs to be increased. 3 lungs initially in the PNAS paper, 2008. And that still holds true. And COPD, we are not there, I don't know if you guys are there, of having that specific profile goes along with a specific genotype. We're not so far yet, but I don't know, you may want to add.
The last short question and short answer, please, not to shorten too much.
Maybe we should talk about this later on, because that's not a short question. Now, the only thing I was going to say is that it's interesting that, and this may be kind of linked to what you are saying, IPF and COPD both happen in smokers. Interestingly enough, when you look at the lung function test in IPF, none of them is obstructed. I mean, the obstruction does not happen. So it seems like, there's a choice to be made. It's either you damage the epithelium in both, but one goes into distraction and the other one goes in excessive repair.
Exactly. And I think the progenitor is the answer.
To me, that's the way of looking at these diseases.
Absolutely.
And that's the future of to understanding the diseases.
Completely agree.
Thank you very much. I don't know if my co-chair has any additional comment. If not, we have now a coffee break. We have 5 minutes less, but we must start sharp at 11.
I need it.